A gene is just a small portion of a single DNA molecule, and it codes for a specific protein. So you can see here that just one small section of DNA is highlighted, and that small section will contain a gene. Now the proteins, if you recall, are composed of repeating units called amino acids. These are the monomers of the polymer protein. And you can see here this polypeptide is composed of 4 single amino acids. Now genes specify the order those amino acids need to be placed in to build the protein, and this is the purpose of our genes, to construct or to provide the instructions for the construction of protein. And it does that by specifying the sequence of these amino acids directly by the code or the sequence of bases within that gene. Now, scientific evidence has shown that information does not come directly from DNA to build the protein, but it must first be mediated through the formation of RNA, and this is called the central dogma of gene expression. Genetic information travels from DNA to RNA and then to the building of the protein from that RNA, what we call the transcript. And the building of that RNA from the DNA in the nucleus is called transcription, one of the first process of gene expression. The second process, actually constructing the protein product from the information contained in the RNA molecule, is called translation, and that happens outside of the nucleus. So let's take a look at the structure of RNA and specifically see how it differs from DNA. RNA is ribonucleic acid, and as you can see in this diagram, it is just a single strand. So here we see the phosphate and the 5-carbon sugar and the nitrogen-containing base that composes a single nucleotide. And what we don't see is that hydrogen bonding with another base and a similar sugar phosphate backbone like we saw in DNA. Instead, it's just this one single strand. As the name implies, instead of deoxyribose in DNA, we have the sugar ribose, which has one more oxygen than deoxyribose. Also, we see, differing from DNA, is the presence of a different base, nitrogen-containing base, the presence of uracil instead of thymine. So in RNA, we see the bases C, G, A, and U instead of T. And finally, because RNA is constructed inside the nucleus and then travels outside the nucleus to be translated into protein, we find RNA in both places, both in and out of the nucleus, whereas DNA is strictly held within the nucleus. Okay, so the first part of gene expression is called transcription, and specifically, that is the process whereby we create a specific kind of RNA called messenger RNA, and that happens in the nucleus. So we need to access the genetic code of DNA to build RNA, and that's why this part of the step happens in the nucleus. And recall that there are many, many genes on a single molecule of DNA. So if we want to express a protein from one gene, we're just going to open up one portion of a DNA molecule, and you can see that here. So only the gene of interest is accessed on a DNA molecule. Now we have another enzyme that builds the mRNA transcript. This enzyme is called RNA polymerase, and it will create that messenger RNA transcript, we call it, a copy of the code. And it does this by complementary base pairing using the free RNA nucleotides A, U, C, and G that are present inside the nucleus. So here we see that one of the strands of DNA actually contains the code for the building of the protein, and the other strand here is just called the template strand or the complementary strand. And so to build the mRNA transcript that will actually contain the code for the protein, then complementary base pairing must take place with the template strand. And in that way, we make a copy of the actual genetic code. But of course, in RNA, we see that there will be a U instead of a T where it was in DNA. Likewise, we know this is RNA because we are just making a single-stranded molecule off of the DNA. Once that messenger RNA molecule has been created and is ready to leave the nucleus, it does so through the many nuclear pores that are on the outside of the nuclear membrane, thus entering the cytoplasm, where it will be translated into protein. Now that messenger RNA molecule is carrying the code for the future protein that will be built from it. We can see the sequence of bases aligned here linearly in this specific example. And that code is arranged in these 3 base sequences called codons. And what we see here is that we have a number of codons in this specific example, coding for 7 different or 6 different amino acids. And finally at the end we see a stop codon down here. We'll talk about that in a moment. So how this works, we have a certain codon table down here at the bottom that we can read the code from. So for instance, this first one is GCU, and we can go to this codon table and look at the first base G and move over to the C column for the second base, and then we see GCU right there coding for alanine. And in fact there are many different codons that will code for just alanine itself. And then we see ACG codes for threonine, GAG for glutamate, and so on. Now down here at the bottom, UAG is itself a stop codon, which tells the process to stop. And we'll see that when we get to the next part of the process, which is translation, in the next video.